Good morning, everybody. First of all, I'm really happy to show you the logo of CPHI Awards that we received yesterday evening for PYCLEAR protection innovation. So I will talk about this innovation today. And we are also happy to have announced yesterday morning a new important partnership with Argo company, one of the packaging leader in eyedroppers. So thank you to them. Okay. Oops. Okay. So some words on PYLOTE first. PYLOTE is a new player in the green tech chemistry process. So we are based southern France, close to Toulouse. And we have many proprietary innovations. And the first prize was obtained in February this year at PharmaPack Europe. So for the same technology innovation. Sorry, I have not pointer. Oh, it's not working, but maybe it's this one. Oh, okay. Okay, I will do with my finger, maybe. And we have an international business development right now. We have launched for the first time this technology innovation in February during PharmaPack. So it's quite young innovation on the market, but already primed twice. And the international development is already in Europe, of course, and also in US and in Asia in the few months coming. So we have many partnerships, ongoing partnerships, and many of them are on CE marking products and also on AMM products. We have also on PYLOTE model, specific PYLOTE model. I will explain what we are talking about, that obtain a successful interest from the investors in January this year. So 2016 was a very big year, is a very big year for us. And we raised 2 million euros for the commercial development. And we are present on different markets, not only pharmaceutical, but also on cosmetics, for example. So it's always growing markets, clearly growing markets, food, for example, and industry. The fundamental technology is called pyrolysis pulverisée. It's a green tech process because we use only water without solvent, and we generate no waste, only microspheres. So this is the small beads, like beads, but it's at a micro size scale. And we have 100% mineral composition, so we don't add any molecules or silver or nothing else. So it's 100% ceramics. These mineral microspheres can be integrated into numerous materials to allow our clients to put on the market final product, greener, cleaner, and safer. So all we are developing in PYLOTE is in this way, to put on the market better products. So one of the interesting things we have developed is that we need no change, no modifications in the supply chain, nor in the final product. So it's remained the same. That looks the same, but we have a huge effect. I will talk about that. So we have different applications, not only for PYCLEAR protection, but with our portfolio. The first one is, of course, to reinforce protection, so to replace preservatives. We have sort of same behavior, performance to consumer products in the food to prolong shelf life and also reduce waste. And we have also innovative technology on the traceability issues, all this kind of final products like oils, arts, and luxe, of course. And we have other technology to improve efficacy, energetic efficacy. So it's done on oven, like you can see there, to increase energetic class of the product. So it's without link to the antimicrobial protection, but it's with the same philosophy and with 100% mineral microspheres. So we have another technology that we present on our booth too this year at CPHI. It's to extend the shelf life of an action, of an performance by controlling the release process. So it's like drug release with 100% mineral microspheres. So you can find there all the PYCLEAR technology under the PYCLEAR trademark. You have the first is PYCLEAR protection, I will talk about that, and PYCLEAR preservation for food industry, PYCLEAR energy for the insulating performance, PYCLEAR authentication, so infalsification, and PYCLEAR duration, like drug release. So we are going to move on the real subject of this conference, and sorry, just to maybe you know, of course, you know the challenge in pharmaceuticals products is to remove preservative, because preservatives, antimicrobial preservative, of course, have different side effects for patients. And many publications, scientific articles show that. And in a worldwide trend, all the health authorities encourage the unpreserved formulation in multi-dose products. So it's true for nasal and eyedroppers, for example. So actually, pharmaceutical labs have two options. The first one is the monodose, and you know that monodose is expensive, it's ecologically not really friendly, and it's not so practical when you have a long-term treatment. And the second way is the called preservative-free multidose. This one are based on the mechanical protection to protect the content of the bottle. So I will come back on this notion. So this increased the number of packaging pieces, so the number of things to produce and the number of materials. They protect only the content, and they have often a silver, it's an antimicrobial substance approach. And today the authorities, like in France ANSES, in Germany BFR, recommend limiting the use of this kind of molecules. So these molecules is suspected to be cancerogens. So au final, we have cost, environmental, regulatory or application issue because it's difficult to used by the other life, for example. So just to take minutes on the reality of the use of this kind of product, you can see by this clinical study we have mentioned there that it makes sense. Of course, the contamination is more often localized on the tip. So it's normal. We can have a contact with skin and with air on the tip. So the majority of the contamination is located there. And less than 15% is located in the residual solution, so in the content. So the technology I'll show you before protect only the content because they put some filters, so many kind of approach, mechanical approach between the tip and the content. So it's not sufficient for us to protect the patient. It's why we have developed PYCLEAR protection. So PYCLEAR protection was developed in 6 years ago for the first time, and we have clearly an interest not only to protect the content, but also each drop we deliver to the patient. So it's the patient first for us too, and we have an answer to solve the full contamination of each drops. so the delivered dose. And we want to, of course, remove antimicrobial preservative because authorities asked for that. And we have no change in patient treatment method, so still use Argo, for example, eyedroppers, so that remain the same eyedroppers. And of course, and it's evident things, we have regulatory safety and stability compliance with PYCLEAR protection. So we have developed microsphere among additive authorized lists like monographies for pharmacopoeia, for example, or USP. So we are today food contact agreements. We are pharmacopoeia European agreement compliant, USP compliant. So it's already done, okay? So it's nano-free. We produce microspheres, no nanospheres. It's clear. It's micro, and we control this parameter. It's silver-free. Like I told you, it's without any additional agent from the ceramics. Okay, so it's 100% ceramics. It's integrable directly to plastics, and we have demonstrated this with Argo, for example. And we have an access to the process like a colorant. So it's master batch, it's commonly used, and it's used like that to integrate PYCLEAR protection in the plastic materials of the packaging. So we obtain a surface effect without migrations. It's not based on something that will release inside the formulation. We have a surface effect and no migration for a full stability of your product. So some pictures. The first graph explains the process, pyrolysis pulverisé process, the green tech process. So we, sorry, I will change my position. We take from there a solution, a water solution, and we obtain at the end of this one-step process, mineral microsphere. So you can see there a micrograph of this microsphere. So it's very microsphere, unagglomerate microsphere, so individual spheres. It's why we can integrate this in plastics without really modification of the process, without modification of the plastic behavior or performance. And you can see there, if we take a micrograph of the plastic surface, all the white spots are microspheres. So it's clearly homogeneous repartition on the surface and also in the volume of the plastic, all the thickness. And its distance between each microspheres as of the same size order of strains, bacteria. So it's why it's so performance. We have between each microsphere sort of microns, 1, 2 microns, depending. So this picture shows the plastics, the microspheres integrate to the plastics, and we again process like a colorant. So with a master batch, a concentrate of microsphere in plastics to be used directly in the actual converting process. So some words on the mechanism, of course. Mechanism. Pyrolysis pulverisé at the beginning leads to specific microspheres. We have some defects on this surface. It's not visible with your eye, it's visible with microscopy. But these defects are interstitial ions and oxygen vacancies. So it's imperceptible defect for you and me, but for the bacteria, it makes the difference. We generate specific spaces with a very short lifetime. So it's nanosecond, so very, very short time. And of course, during this short time life, they don't go through a long distance. They make only few nanometers of distance between the surface where they are generate these oxygen reactive spaces, for example, and we obtain an action at the extreme surface contact. It's why microspheres are never consumed. The microspheres are not delivered into the formulation or into the bacteria. It's not the case. We have a catalytic reaction on the surface, and it's the product of this reaction that allow to kill bacteria and viruses. So we have paper, submitted paper, scientific paper on that. So concerning stability, we have filled many kind of eyedropper, nasal sprays, bottle, and many other things in cosmetics and many others. And we have measured on final and commercial products, so final formulation, different parameters, so droplet size, zeta potential, pH, conductivity, osmolality, ductal contents, etc. And we obtain all the time a stability of the formulation during all the treatment period and all the storage period. So with the ECH stability process and all the things needed for the regulatory file. And we have no impact on the critical formulation attributes. So somewhat on the performance. So the first things we do is to control, sorry, I will go back to this one. We have to control the performance directly on the plastic surface. So on this surface, it's on the tip, on the border, and many things. So to do that, a standard was used. This is the number Japanese standard, but you have also the ISO standard. That's quite the same procedure control. So we take plastic pellets there, and we put strains on the surface, and we deposit strains with a control volume. Okay. So we have standard plastic pellets, and we have the same plastic but with microsphere. So it's called PYLOTE plastic pellets. Plate, sorry. So we do this kind of dilution and other things. So we obtain this kind of activity. So after 24 hours, so we have the control there. It's the standard plastics with 4 log reduction, okay, log concentration, sorry. So 4 of Staph aureus and almost 4 Escherichia coli. And after 24 hours, this concentration of strains remain almost the same on the standard. It's normal, it's not killed. But if we take a look at the result on the assay, on the PYLOTE plastics, you have almost nothing in Escherichia coli nothing, and almost 0 in terms of Staph aureus. So we have an activity, very important activity, in log reduction there, per square centimeter, almost 3 and more than 4 for Escherichia coli. For the nasal application, it's a great news for us. We have obtained compliance with pharmacopoeia in terms of results, because by using H1N1, for example, the grip virus, we obtain a reduction of more than 2.6 log reduction. So it's very important result for the nasal spray products. So this test is done with 24 hours, at 24 hours. But the question is, what happens between 0 and 24 hours? How many time we need to start to kill something? And we make kinetic activity protocols. So after 2 hours, we start an activity, antimicrobial activity. And after 6 hours, we are almost at 2 log reduction. So 6 hours, it's the time, interesting time to fit with the applications, the common applications. So this part was on the plastic surface. Sorry. Okay, you have a bug. Okay. This part is on the plastic surface, but also we have, and I told you, interest in the measurement of the protection of each drops delivered by each kind of packaging. So we have performed trials that you can find on your website. It's a scientific paper already accepted by the reviewers. And we have, it's quickly, we have 2 times per day dispenser contamination, okay? with the 5 strains of the European Pharmacopoeia. Okay? So we put strains on the tip, and we measure not only in the content, but also in each drop we deliver. And we obtain a full protection, where at the same time, in the same condition, the packaging, standard packaging, is contaminated. So it's marketing representation. So I have data for you. And you can see there that after 7 days, you have control and piloted it. So control beginning to be contaminated. Okay? So 300 is the maximum for the countable bacteria. So it's why it's the maximum value. So it's full contaminated. And you have contamination after 28 days. So each day, 2 contaminations per day, you have full contamination where we remain completely protected of each drops. And it was done on different kind of numeration process. So the Pharmacopoeia, European Pharmacopoeia, says that up to 1 month, so 4 weeks, 28 days, you don't need authorization to put on the market multi-dose, for example, ophthalmic. So it's a very interesting result to put on the market a product without authorization needed for that. So in terms of economical benefits, we have a solution adaptable to every kind of packaging, and we have already proved with Argo, for example. And we have an improvement on the supply securities. We have an immediate gain because all this is already done in R&D. No investment needed, because for Argo company, it's without investment in the process, in the peoples, in the materials. So it's full compliance with US, like USP, and Pharmacopoeia, European Pharmacopoeia. Food contact agreement and EcoCert again. Oops. So if I want to resume quickly, we have an innovative technological solution to remove preservatives from solutions, multi-dose solutions, and we have a full microbial protection, 100% mineral without nano, without silver, that's clear. We allow a simple packaging used, so without modification of the treatment method, in the same way, same facility, and no change in use, design, manufacturing, and other things. Maybe it's why we won these 2 awards this year in few months difference, February and yesterday for CPHI award. So thank you. We have a booth to M11 if you have any question that we can't solve today. This talk. Thank you.